So good morning, everyone. So it's my pleasure to present you my presentation regarding molecular diagnosis of myalgic encephalomyelitis, also known as chronic fatigue syndrome, and is distinction from other comorbid condition. My name is Alain Moreau. I'm full professor at the Department of Stomatology at the Faculty of Dentistry and Department of Biochemistry and Molecular Medicine at the Faculty of Medicine at Université de Montréal. I'm also the scientific director of Open Medicine Foundation Collaborative MECFS Center at Sainte-Justine, Université de Montréal, and also the director of the Interdisciplinary Canadian Collaborative Myalgic Encephalomyelitis, a national research network funded by the Canadian Institute of Health Research. Here are my disclosures. So you see the fundings of my different research programs on different fields of complex diseases. And also I'm reporting as part of my role of ICANSEME Research Network, and also my role in a member of the Open Medicine Foundation Scientific Advisory Board. And I'm also participating as a senior editorial board member of Scientific Reports, and I'm the chief scientific officer of a startup called Inception Therapeutic regarding the development of new therapeutic tests and approaches for osteoarthritis. A brief introduction about myalgic encephalomyelitis. So starting with a clinical definition, description of symptoms, and some data regarding the epidemiology and etiology. And also I will cover during the few minutes recent research advances, starting with the development of tools for clinical evaluation, which are often referred to deep phenotyping. And we will explore the role of microRNAs, as well as the exploration of the role of synguanylin phosphodiesterase acid like 3B as a very promising marker for this condition. So often myalgic encephalomyelitis offers several challenges: unknown cause, no cure, and I hope that after my talk you may see some new hope. So one good illustration about what is myalgic encephalomyelitis, like many other complex diseases, is it's like trying to solve the Rubik cubes with changing colors. So the disease is complex, it's a multisystemic illnesses, and also many organs and tissues are affected at different times. So what we know about the presentation and recognition of distinct symptoms is that it's a very disabling in complex illnesses that affect many systems and organs, and people with ME/CFS have overwhelming fatigue that persists for over 6 months or more in adults. And the hallmark symptom of this condition is the post-exercise malaise or post-exertional malaise, in which physical or mental activities leads to a usually delayed and prolonged exacerbation of symptoms. There is no restorative sleep or a variety of sleep disturbances, and either cognitive impairment, often referred to a brain fog by patients, and or orthostatic intolerance are present. About 75% of the cases are triggered by a viral infection, although other type of infection triggered by, for instance, chronic Lyme disease, can lead to ME/CFS. There is a female to male ratio of 4:1, and like many other musculoskeletal diseases, females are more affected for different reasons. You can see on the diagram that is from the US ME/CFS Clinician Coalition handout the different symptoms affecting ME/CFS. In terms of epidemiology, this is not a rare disease. This is a disease that affects a lot of people. So you see some data from the United States in 2015. And in Canada, the disease is in 2019, according to Statistics Canada, almost 500,000 of people receive official diagnosis of ME/CFS. And we know that this is probably the tip of the iceberg. So in Quebec, where I'm working, the number of cases is increasing, and right now we are about 70,000 cases. The prevalence of women between 30 and 55 years old is about 60% of the cases. And of course, this serious illnesses leads to some serious impact in terms of productivity for the society and in medical costs, as you can see on these data from the United States. What we know about the etiology of the disease. First, we know that there are some kind of genetics predisposition. This is what you can see on the upper part of the screen, where this is a UK family over 3 generations, where you can see with the blue dashed figures that the grandmother suffered of ME, and her sons and daughter also were affected. And now the grandchildren, all of them are now affected. So you can see a kind of amplification. So this is a typical multiplex cases where we have proved that there are genetic predisposition. But most of the cases relate to what we refer to epigenetics. So epigenetics will be alteration of your genome without having specific variants or mutations that are embedded in your genetic code. So that's why we saw different viruses, bacteria that can be trigger factors, but also other environmental factors like exposures to heavy metals, arsenic, and other chemicals that can be part of the environmental factor that might trigger ME/CFS over time. Well, we have some kind of onset. A lot of people report to that the disease started with a very bad flu, for instance, or having a severe mononucleosis triggered by the Epstein-Barr virus. Very few people have a full remission at some point of the disease. Most of the patients further progress. And of course, they have much severe cases, so you can be bedridden if you are very, very affected. So, and or housebound, which is also the case of many individuals. And because most of the people suffering of ME/CFS are so aging adults, you may see some complication through different comorbidities like cardiovascular conditions and other metabolic conditions like diabetes that further exacerbate this condition. I learned a lot working with patient partners, and that can be a true challenge for the medical doctor assessing these patients. And this cartoon I like very much because it illustrates really what is happening. The patient expert is the expert of his own illness, and it's quite challenging to at the same time to explain his own problems, to educate the attending physicians about his or her condition, and that reflect this challenge. So as a researchers, I benefit of patients expert to define new approaches to address what is causing myalgic encephalomyelitis. So patients should be seen as a partner, and this is one of the take-home message of this slide. So we developed a first study called Maestro-ME, which is stand for multi-omic approaches to resolve myalgic encephalomyelitis. And the overarching goal of this study or project is the characterization of the biomolecular signature associated with myalgic encephalomyelitis using multi-omic approaches and machine learning models. So, in other words, we try to understand the disease with all the complexity, especially by focusing our research on pathogenic malaise, which is the hallmark symptom of this disease, and that help us to distinguish this disease with other conditions where you may have a very profound fatigue. But in the case of ME/CFS, the post-actional malaise is quite unique. So to do that, and I would try to go through this cartoon, we ask all participants to complete the self-report health questionnaire. We are using 3 questionnaires called SF36, MFE20, which is the multiple fatigue inventory, and the SQ2. This is the POL symptom questionnaire, which is more specific for ME/CFS, to assess each participant about their health condition during the last 6 months. On this cartoon, because most of our participants are at least housebound, we are sending clinical research nurses at home during 3 visits, so which are identified here by V1, V2, and V3. The first visit on V1 is to establish the baseline parameters. And to do that, we are assessing the neurocognitive function using a game on a iPad, which is the brain check test. In 15 minutes, we have a complete evaluation of the neurocognitive functions of the participant. We are also putting the participant on a smart connected vest called Exoskin. So this smart connected vest is measuring in real time the heart rate variation as well as the respiration rate as well as sleep disturbances. So we are asking each participant to wear the vest for at least 20 hours over 24. We are collecting different fluids, so first morning urines every morning, saliva samples, and some blood samples taken at baseline to establish the reference value. So these recordings are performed almost during 2 days, and at the second visit, here you can see on a Wednesday, we are introducing a standardized stress challenge that we call the stress test here. And you can see on the pictures below, this is the application of a mechanical stimulation by putting one arm with an inflatable cuff, and using this cycling of the cuff, we apply a gentle massage. So for most healthy people, they are receiving a therapeutic massage. But for ME/CFS patient, is like asking them to run a marathon. So it's a very difficult exercise for most of them. So again, in that case, we are in real time recording a lot of the changes occurring in terms of the physiology of the participants, as well as we ask them to go through the brain check immediately after the 90-minute stimulation. So this is what T90 refer. And at the same time, we are recording change in the brain oxygen level that we call brain oximetry using near-infrared spectroscopies, where you can see the frontal electrode apply on the front of the participants. And at the end of the 90-minute stimulation, we are also taking a saliva and blood samples again to see what are the alteration of different markers we are looking for associated with the induction of post-exertional malaise. This is also the meaning of the avatar that I'm using. So you see the minion, and because we are safely but surely inducing post-exertional malaise, we are making the participant a bit sicker during few hours, if not few days. So that's why you see the minion is turning purple, which is reflecting that we are inducing a post-exertional malaise, or PEM. And we know that PAM is also developing with not the same intensity amongst individuals. So which means that the PAM can manifest during the 90 minutes or immediately after, or may happen few hours or few days after the induction of PAM. So that's why having this type of longitudinal measurement during a week is quite unique in the world, and that's allowed us to collect a lot of new information about the extent and severity of the PAM and the variability of the PAM between individuals. We expect on the third visit, 5 days later, that most of the participants recovered, and we ask them to complete a fourth questionnaire that we call the DPMQ, which is to report the intensity and severity of their PAM symptom following the application of this standard stress challenge to all participants. So far, we have done the test over 200 ME patients and almost 73 LT controls. So, and the LT control we are using are sedentary LT control without a family history of ME/CFS or fibromyalgia, which is, we think that is the best type of control we can use for that. One great advantage of this test, beside the longitudinal follow-up, is also that each participant is his own control, because we have the baseline value, the 90-minute stimulation, and 5 days later, we have also other values that we can refer. So if the participant is taking different drugs, medications, supplements, we don't really care, because we are neutralizing the effect of the drugs and medication using this baseline versus 90 minutes or versus 5 days later. So this is a quite extensive but unique setup, and all the machines we are using are portable. So that's why we can test participant at home, and we also enrich our database of unique participants that are severely affected, which normally won't participate in many clinical studies because they cannot walk in the clinic or in the lab. So we are going toward them to collect these very precious information. So, for instance, when we measure the brain oximetry changes during the stress test, so this is an example of a real cases. You can see it for this woman of 41 years old. On the y-axis, so you have the percentage of oxygen in the brain, and the boxes. So the first box represent the 5 minutes before we turn on the machine, the inflatable cuff, and the second box, dash box, represent when we turn off after 90 minutes to see if the values will resume to the normality where they were at the initial stage. And the 90 minutes period, you see the real-time measurement. So the upper part is the total oxygen level for the right in brownish, or the left electrode in the dark blue. And the bottom slopes are indicating a very, very important different parameters. So, for instance, the purple curves represent the total hemoglobin, while the red one is the oxygenated hemoglobin, and the dark one or the black one illustrate the desoxygenated hemoglobin. So that's why we can say from the right side of this participant, there is an increase in brain blood flow because the total hemoglobin is increased, and it's a combination of the venous and arterial blood, which is the corresponding to the desoxygenated and oxygenated hemoglobin. But interestingly, on the left side, you can see for the bottom panel of the second diagram here that the brain blood flow is severely decreased, especially coming from the arterial blood, because the total hemoglobin dropped by at least 15%, which represent exactly the drop in the oxygenated hemoglobin. So we don't know yet exactly what is happening here. We know that such a drop may be associated with bradycardia, may associate with hyperventilation, so on some patient, and also some disturbances in the baroreflex receptor in the blood vessels, and often they are asymmetric and often more affected the left side, according to the scientific literature. So we have different profile here, and those profiles are often matching what is happening or what are the effect of the stress test or the induction of PAM on some neurocognitive functions. So we are using a validated neurocognitive test called BrainCheck. Here you see a report, so you can read it. So for each domain, cognitive domain, you have a value over 200. So, and of course, if you got 200 over 200, you are a genius. But most of the people will be around 100, which is the normal population, and we can also generate your rank in terms of percentile compared with the normal population. So you have a global score, which is not really that important, but what is important is the specific changes occurring at specific domain, which allowed us to further understand what is happening. So this is another real case, the ME 196 here, where you can see that there's a pretty normal score at baseline, and immediately after the stress test, you see that the memory, immediate recognition, and delay recognition were severely impaired for both functions. So here, and this is a typical effect seen in ME/CFS patient. So having developing very interesting tools to do deep phenotyping of patients, especially centered around the post-axonal malaise. So that such development allowed us to start to investigate different biomarkers. But where to start? So we decide to investigate first the contribution of small non-coding RNA called microRNA in their contribution in myalgic encephalomyelitis. So, as you may remember from your biology courses, so DNA is our database. So every single cells in our body have the code to make all the proteins that make a human being. But this DNA will be translated, okay, to make what we call messenger RNA. So those RNA contain specific instructions to make the proteins, which are the functional product. And because a bone is not a heart or a heart is not a brain, so that's why the specific instructions that are coming from the RNA are important. And the other type of RNA, so they have the microRNA, so they are very, very small, and they have the property to attach to the messenger RNA at the 3' UTR. So this is at the end of the messenger RNA. And by doing this form of attachment that prevent the decoding of the messenger RNA inside the cell by what we call the ribosomal machinery. So in a layman term, the attachment to microRNA prevent the decoding of messenger RNA and will lead to a reduction or eventually a deficiency of some proteins. And what is very interesting and intriguing is one single microRNA can target up to 200 different genes or messenger RNA. And one messenger RNA may be targeted by more than one microRNA. So the addition may generate some synergetic effect that will really leads to some kind of a protein deficiency or a severe reduction. And if we don't have the right protein amount, we may have the tendency to develop a illness here. So, and also what is interesting with the microRNA is they are responding to different changes in our environment. So it could be about the stress, could be about some hormones, could be the influence of the level of oxygen. So for instance, if you are living on the top of the Himalaya, not only the rarity of oxygen, but also the cold will bring you to a different microRNA signature. Or if you are in the desert at the level of the sea, you will have a complete different microRNA signature just where you are living. So they are more and more studied and used now in molecular medicines to stage diseases, for instance, to stage cancer tumors, to also address cardiovascular diseases. And I will show you data that myalgic encephalomyelitis is no exception here. So we published in November 2020 in Scientific Report a first paper showing a specific profile of 11 circulating microRNA associated with not only myalgic encephalomyelitis, but also in relationship to their symptom severity and disease pathophysiology. So this paper has been recognized among the top 100 paper published in that year in 2020. And also right now you can see that is over 22,000 downloads since November 2020. So what we did is to first, using this panel of 11 microRNA, we developed the first diagnostic prognostic tools of its kind that allowed us, using machine learning, to the possibility to generate cluster of patients. So you see on cluster 1, 2, 3, 4, the way that we have a differential response. So this is the expression of these microRNA that we detect in the plasma after the stress test, after 90 minutes, versus their value at baseline. So for instance, in cluster 3, you can see that all the value increase after the stress test, while in cluster 4, they are decreased. And you can see also on this diagram, all the 11 microRNA are indicated in light blue, and their known or predicted target genes are in green, apple green, and between you see that a brief summary about the pathways or the biological function of these genes. So it's no surprise here that we can see association with mental fatigue, fatigue, physical fatigue, fatigue of muscle, mitochondrial disorder, chronic fatigue syndrome, sleep disorder, other more metabolic pathway like the export of acylcarnitine. So all these functions are known to be affected in most of myalgic encephalomyelitis patients. So there are no random microRNA, they are very specific, and that allowed us to identify additional targets that can be eventually targeted with existing drugs to change and to try to alleviate the most debilitating symptoms. So, beside the association with the disease and how we can understand its complexity, we can also use this profile to predict the therapeutic response. And this is, I think, is a big step where we bring myalgic encephalomyelitis in the have of precision medicine. So this is a cartoon that give you just a simple example with one microRNA, the MYR68193P, which is known to target the TLR3. So this is a toll receptor involved in innate immunity. And this receptor is specifically targeted by ampli-genes, which is an agonist of TLR3 receptor, which has been developed 20 years ago, which is not approved by the FDA, which is only given on few sites in the United States, where about 25% of patients respond quite well to this treatment, while 75% are not responding. And this is exactly what we are seeing here. So we know that about 75% of our participants will have very high level of this microRNA, which means that the target would disappear. And if the target is not there, there is no way that the drug can interact with and have a beneficial effect. So we can do the same exercise with many others microRNA, and that's why I think it's very important for the design of new clinical trials to know to which subgroup of patients should be treated or not with a specific drugs. For instance, we did recently a similar analysis, and we asked ourselves if the microRNA that we use for ME/CFS, where you can see on the panel A, can be used also to understand other related conditions like fibromyalgia. So you can see on the panel B, the same microRNA in light blue, different target, of course, but those targets are known to be associated with fibromyalgia. So we're wondering what is the new signature using the same panel of circulating microRNA toward the prediction of fibromyalgia or not. So as you may know, fibromyalgia is recognized as a rheumatic disease by the WHO, the World Health Organization, since 1992. It's a very common disease, about 2% of the population, but varies by country. Again, female to male ratio up to 8:1. It affects all age group, regardless of ethnic origin or socioeconomic status. The cause is unknown. It's more associated, or more the main component of fibromyalgia is pain, and there is a high sensitivity of some articular pains or muscle pains that can be from diffuse to specific on some joints. There are other overlapping symptoms with ME/CFS, and it's not uncommon to figure out or to observe fibromyalgia as a comorbid condition in ME patient as well. So having this in mind, we use our panel of circulating microRNA to address if there's a different signature in people having only fibromyalgia versus people having both condition, ME or FM, versus the one having only ME. So this is a manuscript that is currently in revision. So, and this is quite important because both conditions are unknown origins. And it's often difficult to address who is suffering of ME or FM. So we start to validate those 11 microRNA in the plasma of FM and ME-FM patients to compare with healthy control as well as ME. So we start with two known microRNA, MIR29A3P and MIR374B5P, because these microRNA were previously reported as strongly downregulated in fibromyalgia patients. So we confirmed that our cohorts were well characterized, and indeed, as you can see on panel A and B, in FM, we hardly detect the expression level of these two known fibromyalgia microRNA. And we, as you can observe here also, by testing ME patient without FM, we find a stronger expression as we did before. And interestingly, when patients suffer of both conditions, we have a kind of composite, so halfway, if you wish, response. So which allowed us to develop a new algorithm using at baseline the 11 microRNA to predict who is suffering of FM versus ME-FM or having simply ME. So that can be a game changer for a lot of patients and physicians to accelerate the diagnosis of both conditions in the context of using now molecular tools. So indeed, if you look for the 11 microRNA, you can see that it's more or less the same picture. So each panel represents one of the 11 microRNA that we developed initially for ME/CFS. And you can see always the same features, which mean that almost no detection in fibromyalgia and a composite expression level in the context of being ME plus FM versus being only ME. The only exception is for this MIR, the 4433A5P, which seems that is not affected by the presence of the comorbid condition. So, as I said, this allowed us to create an algorithm and to test the differences between the standard clinical diagnosis made by family doctors or some specialists. And we did the testing of our own cohort, where we got initially 92 ME patients diagnosed by family doctor or specialist. And we reassessed them using our new molecular diagnostic method. And you can see that from the 92 ME, our test confirmed 52 as having only ME, 31 having both, and finally, 9 of these so-called ME patients were in fact suffering of FM. It's not really surprising because it's difficult to diagnose correctly ME and FM, and due to also the overlap of symptoms, so we cannot blame the attending physicians to be confused, and there is no lab test for both conditions. So that's why we believe that this panel could be of interest to confirm and establish more rapidly the test. It's the same with the background having both. So of course, being both is more easily to confirm because you attach both conditions as a comorbid conditions. And again, you can see the difference between the current clinical diagnostic method versus the molecular ones. I push a bit the envelope because your group is more interested about Ehlers-Danlos syndrome. And as you know, this syndrome is also associated with the Myalgic Encephalomyelitis. So ME/CFS appears to be common among patients with Ehlers-Danlos syndrome. So you can see in A, the same 11 microRNA associated with ME/CFS. It's still in light blue with the targeted gene in green, and the different biological process in yellow, or in yellow. And the panel B, I position some of these microRNA that are targeting genes that are known to be involved in Ehlers-Danlos syndrome. So the genes targeted and circled in red are the genes that are either leading to hypoinsufficiency. So for collagen 5A1, so we know that hypoinsufficiency, so a reduction of the expression level, is sufficient to leads to a Ehlers-Danlos phenotype. So you see a reduction in the ones that are stated in red. And you see that, so which means that elevation of MIR 155P or MIR 29.3P could be part and explain some Ehlers-Danlos-like symptoms in ME patients, as opposed to a reduction in this MIR 28.5P could lead to upregulation of the expression of this SLC 39A13 genes, which is also upregulated in some people suffering of Ehlers-Danlos. So more to come, I would say, but at least now just using these circulating microRNA, we can start to understand why ME/CFS appears to be more common between with patients with Ehlers-Danlos, and that make a lot of sense. So, and I look forward for additional collaboration with you guys, expert in Ehlers-Danlos, to further uh, decode and see the similarities as well as the differences between both conditions. So, of course, we start to decode and decipher each individual microRNA to understand specific symptoms, especially the one that are associated with very, very severe impact on a majority of ME patients. So MIR 155P is involved in orthostatic intolerance or vascular instabilities, and also post orthostatic tachycardia syndrome or POTS, which are very serious debilitating symptoms for a majority of ME patients. So that's why we pay attention. So, and we identify two new mechanisms related to this microRNA that may explain the development of tachycardia or severe tachycardia in ME/CFS patients. So on the diagram A, you see this histogram. So in the y-axis, the expression level of this circulating microRNA in the plasma of patient. So we compare ME patient with ME patient with POTS, and POTS patient without ME, and you see that in both POTS group, you see an elevation of this microRNA as compared to healthy control. We define two novel mechanisms. So one is the elevation of this microRNA, as you can see here, is targeting the key transporter of the norepinephrine called NET. We knew in the past that NET is inactivated due to specific mutation in rare familial POTS cases, although most of the POT cases in the world don't have these mutations. And we know that there is a kind of deficiency in the NET transporter coded by this gene. So one possible mechanism is caused by epigenetic mechanism, where elevation of this microRNA will reduce the total protein and having the same effect that having an inactivated mutation. So high elevation of this microRNA can explain the POTS. The other mechanism is going in the opposite direction. So a severe reduction of this microRNA will lead to an increase in another gene called EZZH2, which is a transcriptional repressor due to the methyl transferase of this protein that will add methyl group, and by doing that, preventing the transcription of the genes. And what is interesting is if I remove the brake, so if I reduce this microRNA, even though that I'm not blocking the NET transporter, I'm activating a very powerful transcriptional repressor. That explains why I have a very similar effect. So, and for some patient having very, very high level of this microRNA, we don't see tachycardia in them. Why? Because if I block the transcriptional repressor, even though that I have a capacity to block the NET transporter, I have so much copy of the messenger RNA that I don't have enough microRNA to neutralize each of them. So that's why the net impact of the NET protein is increased. So that's why it's very interesting to see that it's not just a matter to be activated, to have some kind of high level, but too little is mimicking the same level of having an elevation of this microRNA. And if you go to the far extreme, having a very, very high level, you will prevent it because you will act on the brake, and the brake will be released, and you will have overall more production of the net transporters. So this paper is under review right now. So we will see if the reviewer accept our proposal in terms of this dual mechanism. But I think it's quite exciting because that gives us additional hopes to how can we treat orthostatic intolerance in ME patients as well as in patients suffering simply of POTS. We use a test more recently, our panel of 11 microRNA to predict the trajectories of the long COVID patients. We select non-hospitalized individuals that suffered of COVID, and they develop mild or somewhere even asymptomatic during their acute phase of the disease. But after that, they develop new symptoms that further stay even after 6 months. And with this group, that allowed us, using the panel of 11 microRNA, to predict who will suffer of myalgic encephalomyelitis, fibromyalgia, both, but also additional conditions, so additional long-term sequelae that are not related to ME, like physical and respiratory dysfunction and physical and neurological dysfunction. But as you can see in the distribution of the cases here, for non-hospitalized long COVID after at least a minimum of 6 months of symptoms post-COVID, up to 49% of them develop ME or ME plus FM. So which is in agreement with the other studies that present that. So we are finalizing our studies here, but I think it's quite promising that can allow us to help attending physicians and patients to understand their condition and to act as soon as possible to mitigate and reverse long COVID. Of course, we are not just looking at circulating microRNA. There are multiple different omic approach like proteomic and metabolomics that we are using. Here you have a taste of different markers. Most of them are proteins that are affecting different system in the body. So I've just listed them to see that we are covering a lot of the body. We have over 20, 25 different biomarkers right now. So some of them are more promising than others. And I would like to take the few remaining minutes to cover one of these markers called SMPDL3B. So SMPDL3B stands for the acid sphingomyelase like 3B. We think it's a very promising, not only marker, but target for ME/CFS. You see on the diagram A that this marker is part of the lipid metabolism involving the conversion of sphingomyelin to ceramide, as you can see here. And previous studies, especially the one made by Bob Navio, reported that a reduction of 50% of ceramide production in men suffering of ME and up to 86% reduction in women suffering of ME. So we believe that the key player here in that conversion is SMPDL3B, which is a protein that must be anchored at the membrane level to be active. And in ME/CFS patient, we saw a cleavage of this membrane-bound protein. And this is a cohort that we obtained from colleagues from Norway. We use as a replication independent cohort, and we got the same result with the Canadian cohort. So you see that more soluble SMPDL3B in the bloodstream and more severe the symptoms. So you see a very nice correlation with symptom severity and the level of SMPDL3B. We look also about the influence of age, and we saw that a much higher level on younger patients, which makes some sense because we suspect that estrogens are playing a role here in the production as well as the unspecific cleavage of this membrane-bound protein. And that's explained why at menopause you see a reduction, and also why females are more affected and liberate or secrete more soluble SMPDL3B than male affected by ME/CFS. Interestingly enough, we started to look at our long COVID cohort. This is part of the SCOPIMED study. And we saw also the same phenomenon. So the PAM score is more elevated in the individual having more circulating level of SMPDL3B. And we saw also that there are individuals having the same severity or same severe symptom with a very, very low production. And we suspect for these patients that the problem is not the accelerated cleavage of the membrane bound, but is the lack of production. So there is a reduced production. So all in all, you need to have more membrane bound protein to do the job. And that's why you see this U-shaped phenomenon here. Too little or too in circulation means that there is less membrane bound molecules. And we saw here that affect also some health questionnaires at the cognitive score. As I mentioned before, we know that the membrane-bound SMPDL3B protein is the active form playing a role in lipid metabolism as well as innate immunity interacting with the toll receptor. But we decide to further explore if the cleaved form is truly inactive or is doing something else. So we study the role of soluble SMPDL3B using cellular dielectric spectroscopy, which is described briefly in the panel A. So to summarize this technique is we use wells or plate with wells with small microelectrodes that allowed us to measure precisely a change in the electric currents that is applied. And what the machine is doing is simply measuring or recording the bioimpedance in ohms. So it's a machine that measures the electric resistance, which is modified when the cells encounter a ligand or a specific hormone or whatever we are giving to the cells as a stimulation. So doing this very precise machine, we allow in panel B to measure the effect of one ligand called CC11, which is a chemokine that interact with the CCR3 receptor. We select this chemokine and receptor knowing that SMPDL3B is predicted and demonstrated on the platform biogrids to interact with CCR3 receptor. And as you can see, pure CC11 increasing concentration activate the receptor, and recombinant protein in two different forms. One is tagged with a GST, and the other one is untagged. Both also activate the CCR3 as predicted on biogrid. So we further assess directly using human plasma rich in SMPDL3Bs on soluble version, and we depleted the SMPDL3B with the same plasma, and you see that we almost abrogate this activation. And interesting enough, when panel D, when we block access to CCR3 using a very powerful antagonist, we saw that we promote an activation using a rich plasma with SMPDL3B, suggesting that indeed, even if SMPDL3B, at least the soluble version, can interact with CCR3 receptor, it might interact with another receptor if I increase its bioavailability, so if I reduce the competition by blocking access to CCR3. And this is probably the key receptor we need to discover to further understand why elevation of soluble SMPDL3B is associated with more severe symptoms. Interestingly enough, we study CC11 in the context of this soluble SMPDL3B, and we saw an elevation in ME with POTS versus ME versus LT control. And previously, others are reported recently that elevation of CC11 is associated with the brain fog. So this is a cartoon showing that there's more higher concentration of CC11. And we are suspecting that maybe this is not truly mediated per se by CC11, but maybe by blocking the receptor with high level of CC11, maybe soluble SMPDL2B could do is tricks. And just to add to the complexity, the CCR3 receptor is known to interact with different agonists, antagonists, and CC11 playing both. So if we have a molecules like SMPDL2B soluble can block this one, we may create a lot of different damages that was never predicted before. So that's why we think we are on something that can be interesting and can lead to new therapeutic approaches for ME/CFS symptom severity, like brain fog or maybe even POTS. So finally, I think we are in the fast track of ME research in Canada. We are definitively in a hot pursuit of personalized therapeutic options. So right now, I show you some evidences that we can identify uh, disease using a panel of 11 microRNA to improve patients' diagnosis and certification. And that can help us to get us more insights about the cause and of the key symptoms. We can predict treatment. I give you an example about the one microRNA that may help us to identify individual that could respond quite well to ampli while others won't. But there are also new hopes for other therapeutic approaches. MicroRNA can be also directly at targets. And one of the promising targets, I just show you some example in ME/CFS and long COVID, is SMPDL3B. So at the end of the day, we don't want to create from scratch a new drug, a new treatment, but we want to be in positions to exactly reposition existing drugs to target more precisely the right patient at the right time to have a high impact on them. So finally, I would like to first thank all the participants and their family for their contribution to this study. I would like to acknowledge the support of Open Medicine Foundation, the I Can See Me Research Network, Canadian Institute of Health Research, as well as several private donors for their support. And also a big thanks to my teams, especially the clinical nurses and all the students, very talented young brains working with me on this very complex disease. So thank you all for you listening this presentation and the opportunity to share with you our most recent data. Thank you.